my name is Cláudio Vrisman. I'm a plant pathology PhD student under the supervision of Dr. Miller and Dr. Raja. Our project is about developing tools to help us understand the interaction of the bacterial plant pathogen Erwinia tracheiphila and its cucurbit host by developing bioluminescent strains of the pathogen. One of the strains was isolated from plants in the genus Cucurbit, and it's a more aggressive colonizer of cucumbers and melons, members of this genus. The other strain was isolated from plants in the genus Cucurbit, and it's a more aggressive colonizer of plants like squash and pumpkin, rather than cucumbers and melons. We will look at differential colonization of cucumbers and cucurbit hosts in real time using a sensitivity camera that can detect the light emitted from our bioluminescent strains. We will also study the different routes of entry for the pathogen. By doing this, we expect to learn how the pathogen colonizes its hosts. Once we have a better understanding of that, we may refine management strategies that currently exist.
The transformation of bacterial strains is done using a technique called electroporation. This process allows us to implant bacterial cells with the genes that will cause bioluminescence. For electroporation to work, bacterial cells must first be washed to remove polysaccharide layers around the cell wall. Cells also need to be concentrated to increase the efficiency of the process. Transposon plasmids are DNA molecules that carry specific genes. For our purpose, the plasmid molecules carry the lux operon, a cluster of genes that cause bioluminescence. This is an image of an Erwinia tracheiphila bacterial cell surrounded by plasmids that contain lux genes. Applying a high voltage shock creates tiny pores on the cell wall. The plasmids are able to pass through these pores and enter the bacterial cell. Once inside the cell, the lux genes will be randomly inserted in the bacterium chromosome to do their job, which is to make the bacteria luminescent so we can see its location inside the plant. To confirm transformation of bacteria cells, we grow the cells on media that is amended with a particular antibiotic. Earlier, when we implanted the plasmids with the lux genes, we also implanted an antibiotic resistance gene. If the cells grow on the media as shown here in these pictures, then we can be certain the bacteria have been transformed. Not all genetically transformed strains function the same as naturally occurring wild strains. We first tested transformed strains for growth rates using light emission. Those that emit a brighter light are most comparable to the wild type in terms of growth rate. Since the locations of the lux genes are unknown, pathogenicity also has to be tested to be sure virulence was not affected.
So once the bacterium has been transformed and has shown similarities with the wild type, studies of virulence need to be performed. Here in this small greenhouse experiment, we are showing the wild type inoculated onto cucumbers and the transformed isolate also inoculated onto cucumbers, showing that the transformation did not affect its virulence. This is the system called IVIS, which allows us to visualize the bacteria inside the plant tissue by detecting the light emitted, which will allow us to, in a real time, track the bacteria inside the plant, not requiring a destructive sample and providing a more realistic view of the disease progress. This was just a pilot test, and we can visualize the bacteria traveling in the main veins down to the stem at 1 day post-inoculation. Right now, new cucurbit plants are being grown in the greenhouse to soon be inoculated with the transformed strain, with the objective of studying the progress of the bacteria from different points of entry, as those being the leaf, stem, hydatodes, and nectaries. We would like to thank SCRI for making this research possible. In the future, we will use this technology to look at the bacterial colonization from different routes of entry,
Besides leaf, we will also be looking at stems and nectaries. We will also use this technology to screen potential new molecules and their control of the pathogen. Thank you very much for watching.